Continuing with the third week of development. The changes which take place in the third week are mainly gastrulation, that is formation of the three-layered germ disc, notochord formation, and formation of the neural tube or neurulation. Gastrulation takes place under three steps: formation of the procaudal plate, formation of the primitive streak, and finally formation of the intraembryonic mesoderm. Three areas which are devoid of mesoderm and are formed only by ectoderm and endoderm are procaudal plate, which is now called the oropharyngeal or buccopharyngeal membrane, cloacal membrane, and notochord. Notochord formation also takes place under three steps. First is formation of a solid cord called the primitive notochord, converted into a notochordal canal, neuroenteric canal, and a notochordal plate. This process is reversed to form again a canal and finally a solid cord called the definitive notochord. The notochord is a primary inducer. It not only induces the overlying ectoderm to form the neural tube, it also induces the differentiation of the intraembryonic mesoderm into three parts. From medial to lateral, these are the paraaxial mesoderm, which lies on either side of the notochord, the lateralmost part, that is the lateral plate mesoderm, and in between these two lies the intermediate mesoderm. Thus, from medial to lateral on either side of notochord, the three subdivisions of intraembryonic mesoderm are paraaxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. The paraaxial mesoderm undergoes segmentation to form important structures called somites. The intermediate mesoderm further differentiates into urogenital structures, and an intraembryonic coelom cavity appears within the lateral plate mesoderm. Starting with the paraaxial mesoderm, it differentiates to form cubical segmental masses known as somites. These are present on either side of the neural tube. These somites are triangular structures with a small cavity. They can be further subdivided into three parts. The ventromedial part is called the sclerotome, the lateral part is called the dermatome, and in between them the intermediate part is called the myotome. The fate of the three subdivisions of somites is: the cells of sclerotome migrate medially. They surround the neural tube and give rise to the vertebral column and the ribs. The cells of the dermatome migrate and line the deep surface of the overlying ectoderm, thus forming the dermis of skin and subcutaneous tissue. The cells of myotome will form the striated muscles. The myotomes present in the cervical, thoracic, lumbar, sacral, and coccygeal regions form the muscles of the trunk. Each myotome is innervated by a spinal nerve and further divides into two parts. The dorsal part of the myotome is called the epimere. It is supplied by the dorsal ramus of the spinal nerve and forms the muscles of the back. The ventral part is called the hypomere and is supplied by the ventral ramus of the spinal nerve and forms muscles of the body wall. Apart from these, we have pre-occipital myotomes which form muscles of the eyeball and occipital myotomes which give rise to the muscles of the tongue. In humans, 42 to 44 pairs of somites are present and they are named according to the region they are present in. Example, 4 pairs in the occipital region, 8 pairs in the cervical part, 12 thoracic, 5 sacral, and 8 to 10 coccygeal pairs of somites. These somites appear from day 20 to day 30 in a craniocaudal direction. On day 20, the first pair of somites appears and further 3 pairs are segmented or added each day until the end of the 5th week. Hence, The number of somites gives an idea about the estimation of the fetal age approximately in days. The lateralmost part of the intraembryonic mesoderm is called the lateral plate mesoderm. It extends from the intermediate mesoderm laterally up to the extraembryonic mesoderm. Within this, small cavities appear. These small cavities later fuse to form a large horseshoe-shaped cavity which is called the intraembryonic coelom. This intraembryonic coelom now subdivides or splits the lateral plate mesoderm into two parts. The somatopleuric or the parietal lateral plate mesoderm is the part which is in contact with the ectoderm, and the splanchnopluric or visceral lateral plate mesoderm is in contact with the endoderm. Gradually, this intraembryonic coelom completely separates the somatopleuric and splanchnopluric layers laterally and communicates with the extraembryonic coelom. The intraembryonic coelom thus splits the lateral plate mesoderm into somatopleuric and splanchnopluric layers. Later, it extends laterally and becomes continuous with the extraembryonic coelom. The intraembryonic coelom will give rise to the body cavities, that is pericardial, pleural and peritoneal cavities. The somatopleuric lateral plate mesoderm, which is in contact with the ectoderm, will form the parietal layer of peritoneum, pericardium and pleura, and also the skeletal elements of the limbs. Whereas the splanchnopluric lateral plate mesoderm forms the visceral layers of pericardium, peritoneum and pleura, and also the smooth muscle and connective tissue of the gut, respiratory tract and heart. To summarize, the intraembryonic mesoderm is subdivided into three parts from medial to lateral. These are paraxial mesoderm, intermediate mesoderm and lateral plate mesoderm. Paraxial mesoderm forms somites which further divide into sclerotome, which gives rise to the ribs and vertebra, myotome which forms the skeletal muscles, and dermatome which forms the dermis of skin. Intermediate mesoderm forms the urogenital structures. Lateral plate mesoderm is split by the intraembryonic coelom into somatopleuric and splanchnopluric layers. The coelom gives rise to the body cavities, whereas the layers form the smooth muscle and connective tissue component. The intraembryonic coelom is a horseshoe-shaped cavity. The cranial part of the intraembryonic coelom, which lies in the midline, just cranial to the buccopharyngeal membrane, forms the pericardial cavity. In transverse section, the floor of this pericardial cavity is formed by splanchnopluric lateral plate mesoderm, which will later on give rise to the heart tube. This area is called the cardiogenic area or the cardiogenic plate. The intraembryonic mesoderm, which is present at the cranial-most edge of the embryonic disc, is unsplit by the intraembryonic coelom. This unsplit part is known as septum transversum, and this will later give rise to the diaphragm and liver. The connecting stalk is formed by the unsplit extraembryonic mesoderm. As the embryonic disc becomes oval, the connecting stalk shifts towards the caudal or the tail end of the disc. A small endodermal diverticulum arises from the yolk sac near the tail end of the embryonic disc and extends into the connecting stalk. This is known as the Alintoic diverticulum. Now let us summarize the changes in the third week of intrauterine life. Notochord formation induces neurulation, subdivision or differentiation of the intraembryonic mesoderm, and formation of somites. The intraembryonic mesoderm subdivides into three parts: medial to lateral, paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. The intraembryonic coelom, which is formed within the lateral plate mesoderm, forms three body cavities, that is pericardium, pleura and peritoneum. The lateral plate intraembryonic mesoderm will form three structures: the somatopleuric layer, the splanchnopluric layer, and the cranial unsplit part forms the septum transversum.